them. So with that, I'm thrilled to turn it over now to Dr. John de Groot. Thanks.
Thank you so much for very, very kind introduction, and also for inviting me to present today. This is kind of it's a sort of a bit of a monumental task, because the Society for Neuro-Oncology, I guess I've been a member for about, 22 years now. And every year there are more people who come to the meeting, and there is more research presented at the meeting, and it's thousands of presentations, posters, abstracts, and other sort of ancillary sort of meetings where various things are discussed. So trying to put this into a synopsis in less than an hour, and kind of identifying what are the most exciting, most important things to share it's always a little bit of a challenge. So what you're hearing from me is sort of what I found interesting, but it is only a very, very small slice of the pie of what actually was presented at the meeting. So I hope you find this informative. As Naomi mentioned, happy to answer questions at the end. And of course, always happy to answer questions, later on if something comes up. So, I'm going to start with some disclosures. I do a fair amount of consulting with various drug companies to help with their drug development and their clinical trial designs, and sort of evaluating safety. And so this is my list. And so tonight I'm going to sort of break it down into sort of the newly diagnosed patients. And I say glioma because it may be more than just glioblastoma. It might be also grade 4 astrocytoma included in some of the trials. I don't think we have a lot of low-grade glioma data in the newly diagnosed setting. Obviously, there was the Indigo trial that was presented at ASCO, and that has sort of, I think, passed. And so we're kind of moving on from that. But this is the newly diagnosed group. So the format, what I'm going to do is I'm going to present this as the title. So every different study that I'm going to present today, this will be what it looks like on the first slide. And this is meant to just kind of recognize, the people involved and the authors of the presentation or abstract or poster or whatever it was at the meeting. And then I'll kind of explain a little bit about what the study was about and the results, and kind of like summarize sort of key findings. These are actually available. The abstract is available on the internet for free. And so you can always follow up with that if you have other questions, maybe that I don't address. Okay, so the first study is this phase 1 study. So remember, phase 1 is designed just to evaluate safety. It's not really looking at efficacy, just pure safety. And this is a pretty important study because it's looking at what we call sonodynamic therapy. So sono, such as ultrasound, and then dynamic, just alteration therapy with whole hemispheric low intensity non-ablative ultrasound in patients with recurrent high-grade glioma. So I'm going to go through and kind of walk through what this sort of therapy looks like. So may be familiar to some. There's a drug called 5-ALA, which is actually used pretty extensively all across Europe and also at many centers in the United States to help define the location of tumor when a surgeon is in the operating room taking out a brain tumor. So many of you actually probably had 5-ALA. And this drug, under blue light conditions in the OR, they like shut down all the lights and then turn on a blue light, and they're able to identify exactly where the tumor is so they can more safely take out the tumor, but also get more of it out. But there is a second potential opportunity with this therapy where if you use ultrasound, you can actually make 5-ALA create reactive oxygen species, or we call them ROS. And those reactive oxygen species can actually damage DNA. And if it can damage the DNA, then that gives the opportunity to use this as the sonodynamic therapy. So patients drink this FDA-approved drug. It accumulates in tumor cells, right? It goes straight to the tumor. And then once it's in the cell, then the company has used ultrasound sort of to activate this to release free radicals, these ROS in the cancer cells, damages DNA. And then if the DNA is damaged, and I'm going to say the word DNA damage like about 1,000 times tonight, but that leads to cancer cell death. So this is a very exciting and very novel approach. And so the data from the study, again, phase 1, they found out that, this was very well tolerated. There were no treatment-related death. There were actually no serious adverse events, and no major toxicities. And the median overall survival was very encouraging. So 16 months versus, traditionally 6 to 9 months. So that's very exciting. This has led the company to consider moving into a phase 3 sort of registration trial where it would be randomized, and they're very excited about that. And the other kind of fun note about this company is that they actually have a canine glioma study that enrolls patients as in dogs that have spontaneous glioma. It's very, very effective for that treatment. And I've heard there's actually a dog that flies from Paris to get this therapy. I forget, once a month or once every other month. Fun fact. Okay, so the next study is very interesting, and we're going to talk a lot about vaccines tonight. So this is a personalized neoantigen vaccine. Again, it's a phase 1 trial, so very early stage, just looking at safety. But this is, a very interesting concept. So a neoantigen, neo meaning new, antigen sort of protein that's expressed on the surface of a tumor cell, is really designed as a vaccine to stimulate the immune system to attack this neoantigen, which would be expressed on the tumor cell. So it's personalized because, as we know, there's an enormous amount of heterogeneity in glioblastoma. No two patients' tumors are the same. No two patients' tumors are expressing the same antigens on the tumor cell. And so this is meant to personalize this for each individual patient. I think this is a very exciting, area of research. There's a lot of time that we need, more data, and be able to really know whether or not this is effective or not. But in this very early phase study of just 12 patients, all of the patients did quite well. There were 67% of patients were still alive at 2 years, and almost 60% were alive at 3 years. So this is very, very encouraging in terms of the outcomes. But there was one very serious adverse event for which we don't exactly know what happened. And with just 12 patients, it's difficult to know whether or not this is going to be translatable to the broader patient population, as it were. So the next study to talk about is this personalized precision radiation therapy with targeted dose escalation for newly diagnosed glioblastoma. And the key word here, as we all love to say, is AI, right? So this is artificial intelligence guided treatment. This is not a new concept per se. There have been a lot of studies that have tried to identify, what is the best way to do radiation to a tumor so that you can sort of ahead of time anticipate where the tumor is going to spread and treat those areas more intensely. It makes sense, but it's, as I'm sure you've had conversations with your neuro-oncologist, it's very hard to know where the tumor is going to go next. So this is an attempt to use artificial intelligence, machine learning, deep learning, to try and figure out where the tumor is going to go, so you can sort of more personalize that radiation treatment planning. So this is sort of just a general slide. It's a more of a summary of this concept, not necessarily for brain tumors. Actually, I think this is for lung cancer, but really, this is going to integrate clinical, genomic, and imaging data, and with the hope of kind of integrating all this with this AI to sort of automate tumor and organ segmentation. Right, segmentation is identifying where the tumor begins and ends, and where the normal brain begins and ends, and then sort of making sure that you are providing a margin around that. It can help to expedite sort of that quality assurance process, make sure that you're limiting radiation dose to normal brain tissue, and overall, really just to improve the response to treatment, limit the toxicity and adverse events, and to do it in a very timely manner. So there's lots of things that would go into this, sort of that image analysis. At UCSF, we're doing a lot of imaging research, including with AI, to help us to better predict, based on MRI scans, the boundaries that we were talking about, the critical structures, the normal brain, the infiltrative tumor, and then, to be able to optimize the dose so that you can deliver the highest dose to the tumor and the lowest dose to normal brain, and particularly, getting the dose of tumor to areas where you think this tumor might be spreading. And it can be done, again, on sort of more of a patient-specific manner. And there's lots of potential benefits of this. So could lead to better tumor control, preventing recurrence by more precise targeting, minimizing the damage, as we talked about, and then also sort of streamline that and almost kind of automate the plan, which can speed up time from diagnosis, imaging to treatment. So this particular study, again, looked at the targeted dose escalation of patients with newly diagnosed patients, and it escalated up to 75 gray. So you may remember most patients with newly diagnosed glioblastoma would get 60 gray in 30 fractions. So this escalates up to 75 gray in 30 fractions. So, a little, 25% higher dose. And in this particular study, they saw, improvement of median overall survival from 17 to 24 months, which is very significant. And just to make the comment that within our field, we have used dose escalation in many, many trials without demonstrating a benefit in outcome. So 75 gray, 90 gray. So, more is not always better. But in this particular case, they were able to see a benefit, maybe because of that more precise targeting. So this is very interesting. And I think, we're all excited about how AI might be able to enhance and improve patient outcomes through, trials such as this. So we're going to talk a little bit about niraparib. So niraparib is a PARP inhibitor, and this is a study looking at just efficacy, but also how well the drug gets into the brain of patients with glioblastoma. So PARP inhibitors, so you may have heard of PARP inhibitors. So PARP, PARP is a DNA damage repair enzyme. So the way, as we know, radiation and chemotherapy work is it damages DNA, and if the tumor cell can't repair that DNA damage, then the cell dies. PARP is one of these enzymes that comes in and repairs the DNA, and then tumor survives. So there's a lot of good logic in terms of using a PARP inhibitor. Has to be given together with something that damages DNA, and that would be either radiation and/or chemotherapy. And so, and this approach is successful in other solid tumors. So there's a lot of interest in using this together with radiation chemotherapy and glioblastoma. We've not had a lot of success, and part of that is because we're not sure that we're getting enough drug into the brain tumor. And so that's where this study has been really important. And the way we demonstrate whether or not a drug gets into the tumor is through what we call a window of opportunity clinical trial. And so this is what this is. So really the scenario is to, have a patient who's going to surgery, give them the drug before surgery, and then at the time of surgery, take the tumor out and measure drug concentrations in the tumor. That's sort of the basic concept. Otherwise, we really don't have any good way of knowing if the drug ever even makes it into the tumor, or if it makes it into the tumor at a high enough concentration that it can do what it's designed to do, kill tumor cells or whatever. So the top scenario, number one up here, really is kind of the ideal scenario where you would do a biopsy, and then you use a drug and treat the patient for a week or two, and then take the patient to surgery, and then take the tumor out. And then at that time, you can analyze for not only drug concentrations in enhancing tumor, you can do it in non-enhancing tumor, and then you can also look and see whether or not the drug did what it was designed to do: kill cancer cells, inhibit PARP, whatever it might be. There's a couple of other clinical trial designs. We'll talk a little bit about this second one here when we talk about kind of a neoadjuvant clinical trial with pembrolizumab as an update. And then there's a third type of trial where patients' tissue at the time of resection would be compared to their original tumor, in terms of looking at what the pharmacodynamic or what is the biologic effect on the tumor after receiving the drug. So you can still measure drug concentrations in all three of these different scenarios, but it's a little bit harder to measure what actually happened with tumor cells. So this is demonstrating here that this study demonstrated that the niraparib achieved pharmacologically relevant concentrations in excess of other PARP inhibitors. So this is, definitely sort of the leader, maybe, in terms of the best PARP inhibitor for treating brain tumors. The median overall survival was 20 months, which was more than double what we would normally expect, because we would usually expect something around the order of 12 to 14 months. And GSK, the drug company, has agreed to initiate a global registration trial, phase 3 trial, which I think is getting underway. So very exciting and great to see how we're very carefully evaluating what's the best drug to move forward, instead of just automatically going from phase 1 to phase 2 to phase 3 without this really intense important information. So this is another study looking at tumor treating fields from Optune together with chemoradiation, followed by maintenance TTF with temozolomide in newly diagnosed glioblastoma patients. So this was a phase 2 clinical study, and basically comparing using the TTF alongside of radiation, as opposed to starting it after finishing radiation, when you start the adjuvant temozolomide. So 1 year progression-free survival, 36% versus 19%, but the median overall survival is not different between the two groups. So it's not clear whether or not this actually made a big difference in terms of starting at, with radiation. But the company has started this very large randomized trial called Trident, which you may or may not be aware of, where patients are actually starting the TTF together with radiation, and then being followed compared to just receiving radiation, just receiving TTF in the adjuvant phase. So this is a study out of Scotland, and it's multi-center in Boston. This is evaluating, AZD 1390 is an ATM inhibitor, and ATM is an enzyme that's involved in the repair of double-strand breaks. So really wanting to look over on the right side of the page here. So commonly radiation causes single-end double-strand breaks. there are repair mechanisms that are then activated to repair this DNA damage, and ATM is one of those molecules that would repair that damage. So it makes a lot of sense to combine an ATM inhibitor with radiation. This is then can lead to sort of this accumulation of double-strand breaks. This makes the genome very unstable. The cancer cell can't figure out how to divide, and then it leads to death. So this drug is now gone through sort of this phase 1 study looking at combining radiation with the ATM inhibitor. There were 115 patients treated. 75 had recurrent glioblastoma, 40 with newly diagnosed unmethylated glioblastoma. And it was found to be pretty well tolerated. There was some fatigue, nausea, and headache, of course, which we also commonly see with radiation, but nothing more serious. And they found sort of, what was going to be the safe dose. The preliminary efficacy was very encouraging. It was 12 months for the recurrent patients. And I think, I don't see it for newly diagnosed, but I think maybe the data wasn't mature enough to be able to look at that. But it was promising enough that they really want to move forward. So this is actually taken from a meeting before SNO. So I'm cheating a little bit here. This was in April at the AACR meeting in San Diego, where this data was presented, and you can sort of see, we call this a swimmer's lanes plot, which I love that name. And you can just see, how well patients are doing after starting the medication. And you can see, the very good outcomes, particularly with some of the higher doses of 200 and 400 milligrams. And this has led then for this agent to be added to GBM Agile. And talk a little bit about GBM Agile later on with some of the other results that were presented. But this is a registration trial for patients with newly diagnosed and recurrent glioblastoma. This agent, ATM inhibitor, will recruit patients with newly diagnosed glioblastoma, and that will be given together with radiation. So very exciting to have that join GBM Agile, and hopefully soon have patients starting on that treatment. So this study is looking at this new drug IGV-001, which is an autologous cell immunotherapy in patients with newly diagnosed glioblastoma. So it's a blinded phase 2b study. The approach is really, really fascinating. So patients' brain tumors are resected as normal. The tumor cells are then taken and incubated with an antisense oligonucleotide. And then basically, these are not live cells, and they're put into biodiffusion chambers and radiated. So basically sort of kills the cells that are in this biodiffuser. Those chambers are then implanted into the abdomen. And the whole idea here is that you want to have, again, these tumor cells and their neoantigens, like we talked about, or other antigens, are being exposed to the systemic immune system. And they only do it for 2 to 3 days, and then they take those chambers out. But that gives enough time for our immune system to sort of look at, recognize these cells and these antigens as being abnormal, and to elicit sort of an immune response to that. Their very early data, their phase 1b data, was very encouraging. They had, a group of patients who had sort of the highest exposure and were able to go on to standard of care, living 38 months. And the 2-year survival was 44% in those patients that had the highest exposure. to this vaccine. I'm sorry, this really didn't come out very well. It's kind of blurry. But this is what the clinical trial looks like. So patients with newly diagnosed glioblastoma are then screened and randomized, and either randomized to the autologous cell immunotherapy or to placebo, and then they receive the treatment days 1 through 28, and then following that, the cell therapy, cell immunotherapy treatment or placebo, then they go on to standard of care treatment with radiation temozolomide and adjuvant temozolomide. So this is very, very interesting. The company's name is IMVAX. You may have heard of it. They aren't doing any trials on the West Coast right now because there's an issue with getting the therapy this far west in enough time. But they're working on opening the trial on the West Coast once they have that manufacturing issues resolved. So well tolerated, very promising efficacy signals with improvement of PFS and OS, progression-free survival, and overall survival. There were 15% of patients alive at 5 years or more. And it seems like the adverse events were very, very minimal. And so we're expecting even more updates in 2025 in terms of the safety, but also more importantly, the efficacy. So this is the conclusions from the SNO presentation. Basically, there's no issues with safety, no emerging risks, and it seems like this is very high potential for benefit and low risk. So very exciting type of therapy. So this one I'm a little bit more familiar with. My colleague Naz Majd at MD Anderson and I started working on this in 2018, I think, and she presented this at SNO. So this is a study of a drug called papasertib. Very challenging name to say. Papasertib is a DNA PK inhibitor. So once again, DNA damage from radiation, from temozolomide, and one particular type of repair mechanism for double-stranded breaks is using non-homologous enjoining, and that can be facilitated with ATM, as we showed here, or DNA PK inhibitors. So this is helpful in terms of inhibiting the ability of the tumor cell to repair damaged DNA. And this is some preliminary data in patients' tumor tissues, just showing very high levels of expression, particularly in grade 4 glioma. And that's shown here as well. So grade 4 GBM, very high levels of expression of this protein. And then patients that have very low levels of this protein at baseline do much, much better than those patients who have very high levels of this repair enzyme, as you can see in this plot. So this was a phase 1 study that was designed, so it was in MGMT unmethylated patients. And the idea was to give this drug together with radiation and then to just continue on adjuvant temozolomide afterwards, and sort of a dose escalation, typical phase 1 to find what is a safe dose. And then once that dose was determined, the idea was to actually do one of these window of opportunity trials like we talked about to see whether or not drug was actually getting into the tumor at the levels that we were hoping that it would. The very encouraging survival data, again, for MGMT unmethylated patients. This actually looks very similar to the niraparib study. So, almost 21 months median overall survival in unmethylated patients. Again, typically you would expect to see somewhere around 12 to 14 months. So this is very encouraging. There were only 21 patients. There was a maximum tolerated dose determined. Again, the progression-free survival and overall survival were very interesting and very encouraging. And then I didn't show this data, but we have actually in the collaboration with UCSF, have paired patients' original tumor samples to those that had resection after getting treated with this DNA PK inhibitor plus radiation, and really showing a very robust immune response, and some very interesting genetic changes, which will continue to be analyzed. Doc 1021 is a cell-based vaccination, and it's been tested in a phase 1 trial as in the adjuvant phase for glioblastoma. This is a study out of UT Houston, I believe. And it's a very interesting approach. We've talked a little bit about vaccinations here. So taking out patients' tumor cells and isolating dendritic cells, and then activating those dendritic cells outside of the body, and then putting them back into the patient to stimulate an immune response to those dendritic cells. And the dendritic cells are those cells that are presenting these neoantigens or other tumor antigens, the sort of non-normal antigens. So uses an autologous tumor lysate and mRNA. This is done in newly diagnosed. And one of the really unique features of this trial is that they actually injected these dendritic cells into the deep cervical lymph nodes, so in the neck, which is an area where we believe that immune cells can be primed to then travel into the brain and the brain tumor, just given the location and the proximity to the brain. So this was 16 patients, and it was deemed to be very safe. And the analysis, post-vaccination analysis, demonstrated that there were very high levels of T cells and memory T cells in the bloodstream. So this is really important. I think the next step is really determining whether or not those activated T cells actually get into the brain, and that's, I think, the part of the science that I would like to see to really convince me that this is, an effective approach. So the next one to talk about is a study of autologous, so that's self gamma delta T cells in newly diagnosed glioblastoma in patients getting maintenance temozolomide. This is a study out of UAB with a company called N8 Bio, and so they're developing this sort of immunotherapy with these gamma delta T cells, which are meant to attack the tumor, and they're doing a very interesting engineering feat where they're actually having these tumor cells express MGMT. And the rationale there is that, this is being given together with Temodar, and MGMT we know can help repair DNA damage caused by temozolomide. And so this kind of helps protect the cells, these gamma delta T cells. The treatment is administered to the resection cavity together with IV temozolomide on day 1 throughout each of the 6 maintenance Temodar cycles. And there were patients cohorts receiving 1, 3, or 6 doses of this therapy during those maintenance Temodar cycles. They enrolled 23 patients, but only 13 received treatment. I think in part probably because this is probably a pretty challenging treatment, given that these cells are being injected within the surgical cavity. But in their analysis, they saw 83% of patients exceeded the median 7-month progression-free survival, and feeling like that might be encouraging. So there is continued follow-up on progression-free survival and overall survival. So this is a really fascinating abstract. So this was systemic delivery of low-energy radiofrequency electromagnetic fields for patients with brain tumors. And these EMF are delivered to the patient with sort of a almost like a mouth guard type piece. And this is a company called Autumn. They're based in Brazil. I'm not sure which city in Brazil, but a really, really unique approach. So this Autumn system device emits low energy amplitude modulated systemic electromagnetic fields. And these are done at patient-specific frequencies. Apparently, we all have our own sort of EMF frequency, and this is done to sort of more of a personalized level. This study is interesting. It was a sort of compassionate use program in Brazil. They treated 29 patients with glioblastoma, each 90-minute session every 2 weeks until progression. And patients obviously tolerated it really, really well. But the median overall survival for those 31 patients was 26 months, which is very encouraging, especially given that this is the first time I've really heard of this type of approach, and seems to be very user-friendly, the very low toxicity. Definitely doesn't impair patient quality of life. And so, they're planning more validation studies and doing it in a multi-center trial, which I think is really important to make sure that this is, a real finding. This is a little different. So this is not so much a drug treatment approach, but there is a group called RESECT, the RESECT study group that's looking at different surgical techniques and looking at outcome. And this is comparing 5-ALA guided surgery versus white light surgery. So we kind of mentioned 5-ALA in the beginning and how the 5-ALA when taken either intravenously or orally can go to the tumor and really help the surgeon differentiate what's tumor and what's not tumor, and helps them improve extent of resection. So this was a French multi-center randomized phase 3 study, and basically found a significantly higher gross total resection rate in those patients that got the 5-ALA compared to those in the conventional surgery group. Now, this is actually not completely new. This has been shown in various other studies, and one of the reasons why it's used pretty extensively, at UCSF and other places. And as expected, those patients with a gross total resection did better than those who did not. And they also did sort of a pharmacoeconomic sort of analysis and didn't really add a significant amount of cost to the procedure. So sort of validating the use of this as we have been. So looking in the recurrent setting, there was a lot of talk about GBM Agile at ASCO last year. This is one of the arms of GBM Agile, which is a phase 3 registration platform trial for newly diagnosed and recurrent glioblastoma. Paxilicib is an mTOR inhibitor that was developed at Genentech and then brought forward by another company called Casia. So the structure of GBM Agile, I was editing my slides right before this talk, and I couldn't find the one I was looking for. So this is a kind of an older model, but this is structure, sort of a diagram of what GBM Agile is. So for those who may not be familiar, so it's a platform trial, meaning that it's not just for one drug. It's meant to be for any new experimental therapy that we deem is important to be added to the group. It's adaptively randomized, which means that it's not one patient in each of the arms and then you repeat that randomization scheme. It's actually adaptively randomized such that the arms that are performing best have more patients accrued to them, and the arms that are doing worse have fewer and fewer patients accrued to them. And this is really looking at overall survival. And again, it doesn't matter whether it's newly diagnosed or recurrent. In the newly diagnosed setting, the control arm is standard of care, radiation with chemotherapy and adjuvant maintenance temozolomide. And then in the recurrent setting, the control arm is lomustine. So 50 patients between December 2020 and May 2022. But unfortunately, it didn't meet the threshold to proceed to stage 2, which is actually sort of a randomized phase 3 study. And so it really did not demonstrate a significant survival benefit compared to standard treatment. Median overall survival in the newly diagnosed unmethylated patients was 14.7 months versus 13.8 months. And then 8 months versus 10 months for the recurrent glioblastoma. So unfortunately, that one did not advance, but we have a lot of new therapies that are coming in to GBM Agile, such as the ATM inhibitor, the AZD 1390 compound. Now, so this is a study out of University of Michigan, looking at mycophenolate mofetil to target de novo purine metabolism to overcome radiation and chemotherapy resistance in newly diagnosed and recurrent glioblastoma. So a lot to swallow here. So mycophenolate mofetil has been shown to augment the efficacy of radiation therapy. So purine metabolism, just like really big picture here. So purine is one of the nucleosides. So it's a part of DNA, the DNA molecule. And it's thought that interrupting that metabolism could impair the ability of the cell to repair DNA damage. Once again, DNA damage. And so this data that I'm showing you here is from animal studies, just sort of showing kind of proof of concept. And this group, they showed that it was able to, demonstrate good tolerability, so it wasn't toxic. This mycophenolate mofetil is actually FDA approved, so it's not something new that we're not familiar with in terms of its side effects for patients. And when combined with radiation and chemotherapy, demonstrated an improvement in median overall survival for methylated and unmethylated patients. So very encouraging. And the trial identified the recommended phase 2 dose. And then, they're looking at moving this into a larger randomized trial. So you may all be familiar with this study. For those of you who've spent some time at UCSF, recognize Dr. Jenny Clark. This is a precision medicine clinical trial for adult patients with recurrent glioblastoma. In the interest of time, really, what this trial was designed to do was to take patients who were going to surgery. They would have their tumor removed. The tumor would go through the UC 500 looking for genetic alterations for which there might be a targeted therapy. And so the idea was that once all of that information, the molecular information, there would be a molecular tumor board, really specific for this trial, looking at this patient, looking at their genetic alterations, looking at what drugs they'd received previously, and then coming up with, a 2 to 3 drug combination. It would have to be one sort of cytotoxic chemotherapy, such as Temodar or lomustine or something like carboplatin, and then one or more targeted therapies. And, this is an enormous amount of work. And I think, a tribute to Dr. Clark for all that she did for this study. And we're able to enroll about 30 patients, which used combinations of up to 4 FDA approved drugs, and as I mentioned, one cytotoxic, based on the recommendations of the molecular tumor board. So the most common regimen was lomustine, afatinib, and abemaciclib in 23% of patients. And the efficacy was what we call modest, meaning it was about average. So 9-month overall survival. And I think, Unfortunately, it wasn't more positive. It is pretty similar to what's seen with other studies, but it does show the feasibility of being able to implement sort of a personalized multi-drug therapy approach for recurrent glioblastoma, which I think is a very exciting thing for which, could be implemented in the future. So this is a study out of Duke, D2C7 IT in combination with an FC engineered anti-human CD40 monoclonal antibody. That is a mouthful. So I'll try and break it down and make it a little bit more understandable. So the D2C7IT is an immunotoxin. That's the IT is immunotoxin. And in this case, you can see here, the immunotoxin is Pseudomonas exotoxin. So this is something that if you put it into the cell, would just automatically kill it. And it's linked to an antibody fragment, in this case, EGFR wild type or EGFR V3. So the idea would be that the tumor cell that expresses EGFR would bind to this antibody fragment, and it would take in this toxin, and then that toxin would kill the cell. And then this CD40 agonist is an antibody that actually is designed to activate immune cells. So you're taking one component of the therapy that's going to kill the cell and release tumor antigens or various other molecules, and then you're giving another agent that's going to activate the immune system. And together, the idea would be to really greatly enhance the anti-tumor response. And what you see on the right here, I won't go into a lot of detail here, but I guess this picture is kind of pretty obvious, where you can see, tumors, and this is in mice, have, a fair amount of tumor here in the control group, but those that got the immunotoxin and the CD40 agonist had a really quite incredible response to the treatment. So there was a previous phase 1 study that looked at this and determined that this was going to be very safe. It has to be delivered through convection enhanced delivery, because it needs to get in very high concentrations right next to the brain. This is something that's very difficult to do if you gave it IV. And they treated 18 patients with recurrent glioblastoma, and as of almost a year ago, 83% were still alive, survival ranging from 3 to 14 months. So it's very interesting, very exciting, a really, really novel approach. It tends to be pretty safe, but also challenging, because convection enhanced delivery, putting catheters into the brain, and infusing a therapy is challenging for sure, but very, very exciting. So this is another study. So there was an early study looking at what we call neoadjuvant. So this is giving an immunotherapy or any therapy prior to surgery, with the idea that giving that immunotherapy is going to enhance the effectiveness of the treatment just by giving it before surgery. And so there was a clinical trial run out of UCLA and UCSF and MD Anderson and various other centers, and Dana-Farber were involved, where half the patients got the drug before surgery, and then after surgery, and then half the patients didn't get drug before surgery, but then did get the drug after surgery. And this was, really interesting. We did a study at MD Anderson that looked at this similar approach with a median overall survival of 20.3 months. Very, very interesting. And then there was a randomized trial run out of UCSF, UCLA, and Dana-Farber, where patients were randomized to these two groups. And so this neoadjuvant group, patients survived much longer than those who got it only after surgery. And this was a bit of a puzzle. This trial was not designed to look at overall survival. It was really designed to look at sort of what's the impact on the tumor biology. But nonetheless, there was this survival signal. So the whole point of this trial that was presented at SNO was to to look at this in more detail and see whether or not it could be validated, whether somebody could replicate it. So they repeated the same study. Again, there was a group that got no treatment ahead of time, and then a group that got treatment before surgery, actually in two different doses. And basically what they showed was that in the overall survival group, this cohort here that got drug in the neoadjuvant stage here tended to do a little better than those who did not. Oh, I'm sorry. Actually, yeah, this is the original. This is the original cohort. So when they enrolled additional patients, this black line here, they did not show any difference between those who did not get the drug before surgery. So it kind of, I guess, didn't prove what we had seen previously. So that was kind of unfortunate. So no improvement in overall or progression-free survival compared to controls. So that was disappointing. But I think this neoadjuvant approach, giving immunotherapy before surgery, is still something that is worth exploring. So we have just a few more studies to present. I'm sorry, we're already at an hour. This is in situ vaccination of biopsy-only glioblastoma tumors with tumor treating fields plus an immunotherapy. And the initial results from this study were shared at SNO a year ago. But biopsy-only patients' tumors had significantly improved progression-free survival and overall survival compared to those with maximally resected, suggesting that, not having a complete resection but getting immunotherapy and tumor treating fields could be, a really effective approach to treating patients.
This next study is a retrospective analysis on the safety and efficacy of immunogenicity of 203 glioblastoma patients treated with personalized therapeutic cancer vaccines, or PTCV. This is a study put together based on sort of real-world evidence of patients treated with the CGAT personalized vaccine. And these are the results here. So this is really just recently published, showing that the patients who were treated with the CGAT vaccine had better overall survival compared to sort of a matched control. These would be patients not treated in a randomized prospective study, but patients who had been selected based on some other past treatment. It also demonstrates that not only were patients just overall likely to survive longer with the CGAT vaccine, but also those patients who had a greater immunologic response, so showing an increased immunologic response in the bloodstream, actually did even better compared to those who did not have that same immunologic response. So this study, I think it's important to note, was retrospective. The vaccines were really well tolerated, really not a whole lot of side effects. There were 97% of patients that did develop vaccine-induced T-cell responses peripherally. It has not been demonstrated to happen in the tumor, so this is an important distinction. Median overall survival, the entire cohort was 26 months, and then those that were methylated was 41 months, and those that were MGMT non-methylated was 23 months. And I just, again, want to emphasize this is not a prospective randomized blinded trial. This is retrospective, and honestly, the results from this are difficult to interpret because, it's really a very select subset of patients who are able to travel to Germany to be able to pay, hundreds of thousands of dollars to get this vaccine. And I think that these results just should be sort of taken with some consideration of these potential major biases. So the next study is a phase 1 randomized perioperative trial of vorasidenib and ivosidenib and IDH mutant diffuse glioma. These are updated results from a trial that was started several years ago. And of course, this is in the context of vorasidenib being approved for IDH mutant grade 2 astrocytoma back in August of 2024. And so this, really, I don't have any additional slides on this, but in summary here, the studies showed that both vorasidenib and ivosidenib were compared in studies of grade 2 and 3 non-enhancing IDH mutant gliomas, IDH1 mutant gliomas. They received treatment for 4 weeks before surgery, and then the option to continue after surgery. And then by September of 2023, 46% of the vorasidenib and 20% of the ivosidenib patients remained on treatment, with the median postoperative treatment duration of 44.7 months for vorasidenib compared to 24 months for ivosidenib. So, I think these data are really, really critical. I think it demonstrates why the company moved and went forward with vorasidenib compared to ivosidenib due to, this much greater PFS benefit and overall response rates. So, very exciting study.
I think we're almost to the end here. This is a very interesting study, and this is really just about the diagnosis and the biomarkers within the tumor. So this gene called CDKN2A, this is something that we know can upgrade tumors from grade 2 to grade 4, but this study sort of looked to see what its prognostic implications were for patients with IDH mutated tumors. And this is a single center study, and they looked at, previous cases of patients in their tumor bank and found that 24% of previously diagnosed glioblastoma reclassified as IDH mutant astrocytomas. And those reclassified patients, about half had CDKN2A deletions, which definitely is a negative impact on survival. And you can see that here. I won't run through the numbers, but if it's a glioblastoma with this deletion, it's 15 months median survival. If it's IDH mutated with this, it's 19 months. And then if it's IDH wild type without it, 22 months. And then the best group, IDH mutant without CDKN2A deletion of 33 months. So I think this is really very important. And, this is this sort of a description of how we diagnose and classify all of the different types of glioma. And the most important sort of differentiating factor is the IDH mutation status. But then, as you can see, if you go into the ATRXP53 mutant group, you're getting into the astrocytoma group. And that's where the CDKN2A deletion testing is just so critically important, because that is enough to classify the tumor as a grade 4. So just in summary, again, I apologize for talking so much, but there's a lot of really exciting data. We talked about sonodynamic therapy, neoantigen vaccines, this AI-guided precision radiation therapy, multiple studies with tumor treating fields. And then, as I mentioned, a lot of DNA damage repair inhibitors. We talked about PARP inhibitors. We talked about ATM inhibitors. We talked about DNA PK inhibitors. We probably talked about some other ones that I'm not remembering. And so a lot of excitement in that area as well. So, I think it's just important to state a little bit of the obvious here, but, this is one of the most difficult diseases to treat. We have lots of different drugs in development. As you can see, like I just presented a tiny handful of what was presented at the meeting. Drug delivery continues to be a really big challenge for us. And more companies are developing drugs with better brain penetration, which is great, particularly for brain metastases. But, we're also using new technologies to help improve delivery, such as focused ultrasound or convection enhanced delivery. just a comment that, retrospective studies looking at what's happened in the past and sort of pulling that data and analyzing it in aggregate and comparing it to something that may or may not be relevant is not a way to actually prove that something works. And so for any of these studies, particularly the phase 1 studies, really larger studies and particularly randomized studies are important to prove efficacy. Thank you for your attention, and I'm happy to answer any questions.
Thank you so much, Dr. Groot, and thank you. There's a couple people that have submitted questions. Okay, the first question is, I'll read it out to you. I recall hearing fairly unimpressive results regarding efficacy of Optune. Is there newer data with more encouraging results?
No, it's a great question. I think, I think the company has done a really great job of trying to really understand the mechanism of how the Optune works. And in doing so, has really come up with some really interesting ideas in terms of making the efficacy even better. And so one of the studies that they're looking at is sort of based on one of the trials that we talked about earlier, where they, these biopsy-only patients that got the treating fields together with immunotherapy. So they're now starting a larger study in newly diagnosed patients where they would get tumor treating fields with immunotherapy in addition to all the other upfront standard of care treatment. And I think that's really great, and hopefully that will lead to additional advancements. But at least it looks like now the adding it to at the time of radiation, may or may not be effective. So hopefully that answers the question.
Thank you so much for that. And I will read out this other question as well. And this is a question about TTFs. And if you could please give more examples of TTFs.
So more examples of tumor treating fields, I guess.
Yes.
Yeah. I mean, I know, actually, I don't think I can answer that very well. I know there's another company, maybe even a couple of companies that are working on similar types of devices. But the one from Autumn, that one with the electromagnetic fields is kind of similar. It's obviously different. It's not a helmet. It's just, having sort of like a mouth guard that emits electromagnetic fields. But yeah, I think I don't know of any of the results from some of these other devices that are currently in development.
And maybe some people aren't aware that TTF is the same as Optune, tumor treating fields. Yeah.
Right.
Optune is a type of tumor treating field.
Thank you. Yeah, and then I see the question about asking Dr. Aghi about a pre-surgery immunotherapy. So that's really, that was the one that we looked at with the pembrolizumab or Keytruda, where the original study that we published showed that survival benefit, but then when tried to validate it with a second cohort, it didn't look like it extended overall survival. So this neoadjuvant approach is very well established. It's actually used in a lot of different solid tumor types. So, for example, melanoma patients, if they need surgery, will frequently get a neoadjuvant, pre-surgery treatment, sometimes even for several months prior to going to surgery, and that has been shown to be beneficial for outcome. Yeah, so most of what we talked about today was about glioblastoma.
Yeah.
Some of it was, included patients with grade 4 IDH mutant astrocytoma, but pretty much the majority of what we talked about today was for GBM. But, to answer a little bit more broadly, I mean, what we learn in GBM can definitely, potentially apply to other tumor types. regardless, whether it's a IDH mutant tumor or even other types of tumors. Information about drug penetration into the brain is a universal interest across tumor types. What it does to the tumor is of universal interest. And using combinations of treatments like the DNA damage repair inhibitors. I mean, radiation works in one way pretty much across tumor types. So having those combination data could be helpful in designing other studies. Yeah, so the doses of ultrasound that are being used right now are at low frequency and kind of low intensity, so as they disrupt the blood-brain barrier, but they're not at an ablative dose. So a long time ago, several decades ago, people tried to use very high intensity ultrasound to sort of burn the tumor and kill the tumor, and it led to some serious bleeding problems. And so that has now been sort of abandoned in a way. And we're just now using it to disrupt the blood-brain barrier, which lasts for about an hour, maybe 90 minutes, and allows us to deliver drugs during that opening.
In last year, I think, was when the IDH1 drugs came out, and there was such great excitement. And did you see any new phase 3 that looks like things are getting close to getting approved, or was this just not the year for that? And we just have a lot of very interesting phase 1 and 2 drugs that will take a little while longer to come to see if it works.
Yeah, I think that's a good assessment. There were some drugs that were in these registration trials for phase 3 that failed, unfortunately, like the Paxlovid. But most of what was presented was sort of that earlier phase where there's very interesting things, but still need to be proven. I think you're right. The IDH inhibitor story is very, very exciting. And with its approval last year. But there are also other companies looking to develop their drugs in that space. So, maybe there'll be new IDH inhibitors and more than just one as a possibility. And then the other is, of course, there are potential combinations. So trying to use IDH inhibitors with other drugs for these, the more aggressive grade 3 and grade 4 IDH mutated tumors. Yeah. I mean, it's a great question. I honestly wish we understood that process better. Why is it that somebody can get, radiation chemo and not have a recurrence for 5 years? And then somebody who looks exactly the same to us has a recurrence at 1 year. Like, why is it so different? And they got the same treatment. We call the tumor the exact same tumor type. And yet, the biology and the behavior is so different. And we've actually looked really hard to see what, is there a genetic change in the tumor that's making it more aggressive? And so far, we have been very unsuccessful finding something that does that, which then begs the question, is it patient related, right? And that I think is kind of fascinating to think about. And what is it about what is it that makes one patient different than the other? And I think it's an area that we need to explore.
Well, this sounds great. Does anybody have anything else? So I think we should thank Dr. Groot for coming to our group. Thank you so much.